Thank you, Amy. It's my pleasure to introduce Dr. Richard Kaufmann as today's Grand Round speaker. Rick is a Pittsburgh native. He received his MD from Washington University School of Medicine in St. Louis, and he also trained in clinical pathology and transfusion medicine at WashU before relocating to Boston. Rick is now the medical director of the Brigham and Women's Hospital Transfusion Service and an associate professor of pathology at Harvard. He has pursued a variety of clinical research interests in the field of transfusion medicine. You'll hear of one today. He also serves as editor-in-chief of Transfusion, a post that was proudly held by my predecessor, Jeff McCullough, for many years, and chairs the U.S. Blood Products Advisory Committee. So it's with great pleasure that I introduce Rick. He's going to talk to us today about severe CD4 positive T-cell lymphopenia that's not observed in frequent platelet pheresis donors collected on certain platelet platforms. Rick, please take it away.
Oh, thanks a lot. So it's really a pleasure to speak to you today. And let me share my screen. So, this is a weird time. Hopefully I can give everybody a break. I've been, kind of white knuckling it through the election and the pandemic, but I hope to talk to you today about something new to worry about. So this is kind of a bedside to bench project that I've been working on for the last couple of years. So my title is Severe CD4 Lymphopenia in Frequent Platelet Donors. And I have no disclosures. So just to start, the vast majority of platelet units that are used for preventing or treating bleeding are collected by apheresis, or what we call platelet pheresis. And I'm just going to quickly show you how that's done. So a volunteer donor will undergo a brief physical examination and then has to pass the donor history questionnaire. And then what's done is to access either one or both of the donor's antecubital veins, and using single-use sterile tubing, we connect the donor to an apheresis instrument like the one that's shown on the left. And whole blood is drawn out of the donor by the instrument, and there's a centrifuge in the bottom that will essentially separate out the components of blood by density. So red cells having a lot of iron go to the bottom, plasma goes to the top, and the apheresis instrument can siphon off platelet-rich plasma, and that will end up in a bag at the back, and then the red cells are actually returned to the donor, and the donor is essentially maintained at the same volume during the procedure. So it's sort of a slick process, and it's possible in a single donation for a donor to donate 1, 2, or 3 apheresis platelet units, each having usually in the range of 4 × 10 to the 11th platelets. So a good sort of normal adult dose of platelets. And there are more than 1.2 million of these types of donations that are done every year, just in this country. So apheresis technology to collect platelets became available in the 1980s. And right at that time, particular dedicated individuals started donating a lot. And so there started to be concerns raised that in using this new technology, were we in any way hurting donors? That is, by people donating platelets frequently, were their platelet counts being lowered to where it might be dangerous for them in that they might be at excess risk for bleeding? And another question that was raised was, well, the system's not perfect. Are we removing white cells? And could that potentially cause the donors to become immunocompromised? And so this was a paper that Ron Strauss, a longtime transfusion medicine specialist at Iowa, published in 1981, called The Safety of Weekly Platelet Pheresis, Effect on the Donor's Lymphocyte Population. And so they did a study where which was a prospective observational study where they had 10 healthy volunteers, just healthy men who donated basically once a week for 12 weeks. And they measured the donors' lymphocyte counts. And so the plot that I'm showing here is their main data. So what they did was for each of the donors, they had them donate 10 times, and then they calculated the mean lymphocyte count for each of the donors across the study period. And then for each of the time points shown on the x-axis, week 1, week 2, week 3, and so on, what they've plotted is the change in percent lymphocyte count, higher or lower, relative to their mean count across the entire study period. And they saw a definite trend that was statistically significant, although not thought to be clinically meaningful, where there was overall about a 22% drop in lymphocyte counts. So in 2008, Ron Strauss published this. So during the interim years, the technology for doing platelet pheresis collections got a lot better, and Ron Strauss wrote that donor lymphocytes collected or lost during platelet pheresis are so few that the risk of significant lymphocytopenia and or immune dysfunction is nil. So this was a problem that seemed to be a thing maybe with the earliest generations of apheresis instruments, but everyone kind of forgot about it. And there was even a time when the FDA required donor centers to keep an eye on lymphocytes, that is to monitor them, but they took that out of the regulations at one point, just because it seemed to be a non-problem. And that's where things remained until one day in 2017, when I came into the blood bank and I got a message that a donor had left a voicemail saying that he'd been diagnosed with AIDS. And I called the donor, and this was someone who was a frequent platelet donor in our hospital-based donor center. And this person had donated platelets by apheresis 212 times over the last several years. Each time, of course, the donors are tested both by nucleic acid testing and by a serologic test for HIV. And he was, of course, negative 212 times. I called the donor, and it turned out that he felt fine. He had volunteered to be a research subject in a study that was being done at the Cohn Institute in Cambridge, which is a place that studies HIV. And they had measured his CD4 count, and it was 186. And so a CD4 count below 200 in people that are HIV positive is diagnostic of AIDS. The normal range for a CD4 count is approximately 500 to 1500 cells per microliter. So his was 186, but like I said, he didn't have HIV and he didn't have AIDS. He told me that he'd gone bicycling 20 miles the day before. And I told him that I had no idea why his CD4 lymphocyte count was so low, but if he wanted, he could certainly come to our hospital and speak with a hematologist here. And in the meantime, with his permission, I called the Cohn Institute to get a little more information. The person that I spoke with at the Cohn Institute mentioned that there was one other time when they had seen someone who was HIV negative, but also had a really low CD4 count. And the person I spoke with thought that person might have also have been a platelet donor. So I thought that was really strange. And so that weekend, I asked our donor center, which had been doing a mobile drive and collecting some platelets on site, to measure a CBC with differential on every donor that weekend. So normally, blood donor centers really just want to see the minimal data that is required for us to collect blood products. And in our case, we just wanted to see hemoglobins and platelet counts. And so even though a CBC with an automated diff was done on every donor, that data was just suppressed and we never got it back. And that's actually how most donor centers, Red Cross and so on, function. Anyway, it looked like there was a signal for a lower CD4 count, a lower lymphocyte count, lower total lymphocyte count among the platelet donors compared to the whole blood donors. And so on that basis, I got in touch with a guy named John Gansner, who was a hematologist who ended up seeing our index donor, and we decided to do a study. The question was, can plateletpheresis cause lymphopenia? And so we decided to do a cross-sectional study of apheresis platelet donors, and we collect all of our platelets on an instrument called the Trima XL instrument. So at our donor center, we have a group that they call themselves Team 20. So these are people who donate the maximum 24 times a year, and if they're in the range of donating at least 20 times a year, then they get this Team 20 credential at the annual donor brunch. So we recruited 20 of these individuals. As a control, we identified people who were brand new apheresis platelet donors, and they may have donated once in the past, but basically had donated something like 1 or 2 donations per year. And then we decided also to look at an equal number, 20 individuals who fell in the middle, so had donated 3 to 19 times per year by apheresis. And this was all we did. So at a single donation, we got consent from these individuals, and we collected a tube of blood for CBC with differential and also for CD4 and CD8 cell measurements. And we also harvested this plastic cone called the leukoreduction system chamber. I'll talk about more a little later. And it's a system that's used in the Trima instrument to try to prevent white blood cells from ending up in the bag of platelets that's collected at the end. Because one hypothesis was, well, maybe we're just losing a lot of white cells in the system, and this would be an obvious place to look for them. So we found that frequent platelet donation using the Trima is in fact associated with severe lymphopenia. And this was the most important piece of information that we found. So here I've plotted the CD4 count, cells per microliter, for each of the donors in the study. And they're grouped, as you can see, along the x-axis into those 3 groups of donors. So 1 to 2 were the new donors. The people in the middle had donated 3 to 19 times in the past year. And the people on the right had donated 20 to 24 times in the previous year. And you can see there was a clear dose response. That is, the people who donated more tend to have lower CD4 counts. There were 0 individuals in the 1 to 2 donations group who had a CD4 count below 200. There were 2 out of 20 individuals, or 10%, in the 3 to 19 donations group of people who had a CD4 count in the range that defines AIDS of below 200. And we were surprised to find that 6 out of 20, or 30% of our Team 20 donors had CD4 counts below 200. So the blue on this plot, the blue symbols means that a particular donor had donated 20 or more times in a single year at some time in the past 20 years. And so you can see that the people who were really low in the 3 to 19 group had all at some point been part of Team 20. So the CD8 counts showed a similar pattern. And again, there seemed to be a dose response curve. The low end of normal for CD8 in our lab was 125 cells per microliter. There were no donors in the 1 to 2 donations group that fell into that group. There were 3 in the 3 to 19 group, and it was something like half of the frequent donors those in the 20 to 24 donations group, who fell below the normal range for CD8. Now, one of the limitations of this study is that those 3 groups of donors that I showed you, the 1 to 2 donations, 3 to 19, and 20 to 24, they weren't all the same age. So many of the really dedicated donors in the US, the people that donate every 2 weeks essentially, they're retirees. So they have time to donate that frequently. And so the median age for the donors in the 1 to 2 donations group was 37, whereas it was the median age was 60 for those in the middle group of donors, and it was 62 years old for the Team 20 donors in the 20 to 24 donations group. And so, as I'm sure you know, thymic output does decline by age. This was shown in several different experiments. This is one looking at TREX, which is kind of a somewhat, at this point, old-fashioned way of looking at new T-cell production. So this is T-cell receptor excision circles that get produced when the T-cell receptor gene undergoes rearrangement as T-cells develop. And you can see from these box and whisker plots that there is a decline in output from the thymus. Starts quite early, actually, but goes down over time. And by the time that you're in your 70s, there may not be any new production of T cells by the thymus at all. The overall T cell counts, though, don't decline so dramatically. There aren't great data for this, but it looks like there's a trend, but it's relatively modest in total T cell counts. And I think how this is controlled is not perfectly understood. So we know EPO is critical for maintaining your hematocrit, TPO for your platelet count. For lymphocyte counts, it's, I think, less well understood. IL-7 and IL-15 seem to have a lot to do with it, but the mechanism of how, what the thermostat is, sort of how your lymphocyte counts are regulated, is a little less clear. We did a multivariable model of our data. I won't spend a lot of time on it, but just to say that it looked like there were independent effects. So that is frequent donation being in the 3 to 19 donations group or being in the 20 to 24 donations group, as shown in the blue rows, definitely contributed to having a low CD4 count. And there was also independently effects of age, and it wasn't possible to entirely tease these apart. We looked at cumulative donations, that is donations over a lifetime. And it turned out that the people who had the really low counts, shown as below the CD4 count of 200 per microliter, the horizontal dotted line, had all donated at least 170 times over the past 20 years. And you didn't really start to see much of a drop in CD4 counts until you had donated more than 50 times, which is denoted by that vertical dotted line. So there seemed to be some kind of cumulative effect of this. We looked at just regular blood counts in these frequent donors. So here, the donors that were in the 1 to 2 donations group are shown in blue. Those in the middle group, 3 to 19 donations in the past year, are shown in red. And the frequent donors, 20 to 24 donations in the past year, are shown in green. And you can see on the left, there were no differences really in total white count. In the middle, there weren't really any major differences in hematocrit, and the platelet counts, likewise, shown on the right, were all about the same in these 3 groups. We did see a difference in lymphocyte count that roughly correlated with what I showed you before with CD4 and CD8 counts. So one question you might ask is, well, how are the total white counts the same if the lymphocyte counts were lower in the frequent donors? And it turns out that the monocyte counts were actually higher in the 20 to 24 group, and that kind of balanced things out. We also did a lot of more detailed analyses of lymphocytes in these donors, looking at different flow markers. And we found a relative decrease in CD4 naive T cells in the frequent donors, as shown here. And there are relatively more CD4 effector memory T cells in the frequent donors, as shown on the right here. I don't know exactly what this means. This pattern, though, is at least reminiscent of something that's called lymphocyte induced proliferation, or LIP. And this is something that's been described in mice. And I'll just show you this really briefly. So if you take a normal mouse and transplant naive T cells from that mouse into an irradiated lymphocyte depleted recipient mouse, the lymphocytes that you transplant will undergo sort of this slow proliferation. And the odd thing is they'll take on a memory-like phenotype based on their surface flow markers. even though they've not been exposed to antigen. And it's believed that, well, one hypothesis anyway, is that there's relatively more IL-7, which appears to get produced kind of at a constant rate, but there's relatively more IL-7 for each cell. And this kind of tilts them toward making more of a memory-like, having more of a memory-like phenotype. And so, I don't know, we wonder if maybe a similar thing, some lip is going on in our frequent platelet donors. So what is the mechanism of platelet-pheresis-associated lymphopenia, or what we call PAL? We didn't really set the study up to try to figure out the mechanism. But one obvious hypothesis, as I mentioned earlier, was that we were simply physically removing lymphocytes from donors as they did these repeated apheresis collections, and maybe that was it. There's if indeed that is the mechanism, I'm sure there's more to it. Maybe they have a different sort of set point eventually. But anyway, this is how this lymphocyte reduction system works. It's a little plastic cone, and plasma kind of flows through it from the bottom to the top, as shown there. And what happens is platelets kind of form this mass at the top of this upside down cone, and platelets kind of percolate out through the top and end up getting collected. Meanwhile, white cells are left behind and are trapped in the cone. So it turns out that the Trima LRS extracts 15 to 20% of circulating lymphocytes and monocytes for each donation. So those data are shown here. And we used essentially the same formula that's used to calculate stem cell collection efficiency when we collect hematopoietic stem cells from donors who are undergoing apheresis for auto or allo transplant. So as you can see here, in the cones, we found lots and lots of lymphocytes and monocytes, very few neutrophils and eosinophils. There was actually something like a 10% basophil loss in the system. Do lymphopenic donors have normal T cell diversity? So as I'm sure you'll remember, that the T cell receptor gene looks like this. It's very reminiscent of the immunoglobulin genes, where there's this combinatorial rearrangement to generate lots and lots of different specificities of T cell receptors that can recognize a really wide array of peptide antigens in the context of MHC. You end up with a protein that looks like this, and everybody has a really normally a really sort of broad array of variations on this. So we did something called immunoseq. We took a bunch of samples, sent them to this one company in Seattle, and their technology has essentially replaced the TREC analysis that I talked about earlier. So what this company does is take DNA, and by next generation sequencing can give you an idea of the diversity of different T cell clones that are in a large sample of T cells. And as you can see here, when we looked at the 1 to 2 donations group, the 3 to 19, and the 20 to 24 donations group, we didn't really see any difference in terms of their T cell receptor diversity. So that at least was a bit reassuring, in that even though many of the donors in the 20 to 24 group had really low T cells, at least they had still a broad specificity and hadn't sort of focused down to a small number of T cell clones. We asked, do lymphocyte counts return to normal when donors stop donating? So we assumed that they would, come back up as soon as you stop doing regular collections. What we did, and this was led by Mandy Ramani, a former fellow of mine, was to look at people who used to donate as part of Team 20, but then for whatever reason stopped donating. Some of these individuals decided it was too much of a hassle to drive in and park in Boston. We had some who were deferred from donating. They might have had 2 false positive hepatitis B core tests, or actually we lost some when the US switched to only collecting platelets from women who did not have anti-HLA antibodies as a trolley reduction approach. So what Mandy did was to send letters to 50 former frequent donors, all people who had donated at least 20 times in 1 year. 15 of them agreed to come in and get tested, and we just checked their CBCs and CD4 and CD8 counts. And this was her main data. So she found that compared with our current platelet donors, the former platelet donors had a slightly higher median CD4 count, but there were still a couple who, even though they hadn't donated in at least a year, had CD4 counts that were below 200. So this person had stopped donating for 2 years and the count was still below 200. And this was actually our index donor, who by this point had stopped donating for over a year. And so this was a little bit worrisome. We don't really have longitudinal data on anyone except a little bit from the very, very first donor that I mentioned. So you can see in the gray plot on top that his total white count remained relatively stable over the year that we followed him. His CD4 count did seem to be coming back up, but it wasn't happening fast. So after 400 days, it was still below 200. So if there is recovery of CD4 counts, and I think that there is, it's not immediate. It takes some time. We asked, is platelet-pheresis-associated lymphopenia platform specific? So I mentioned before that we had been using the Trima Excel with LRS system to remove white cells from the product. The main competing instrument is something called the Fenwal Amicus. Works essentially the same way, except that it doesn't have an LRS. So it keeps the white cells out using a different mechanism. And in fact, per the manufacturer, the white cells should all get returned to the donor. And so we did a preliminary study of frequent platelet donors collected on the Amicus instrument in collaboration with Mona Papari at Vitalant in Chicago. So at her donor center, they collect a lot of platelets using Amicus with no LRS. We took 30 donors who had donated 20 to 24 times on this instrument per year, measured their CBCs and CD4 and CD8 counts. And this is what we found. So this is showing CD4 counts from these frequent Amicus donors and CD8 counts from these frequent Amicus donors on the left and right, respectively. One thing that was a little surprising is that although we saw 0 individuals among these 30 frequent Amicus donors with platelet counts that were as low as we'd seen from the Trima donors, that is below 200, there were 6 out of 30 that were below the normal range for CD4. And I'm not sure what that's about. At this point, we don't know if there's a milder lymphopenia effect of donating frequently on the Amicus instrument, or if the normal ranges are just not quite informative enough because maybe they didn't include enough elderly individuals, which may really be it. This may just reflect an age effect. I don't think we have any idea at this point. And there was one individual who had a CD8 count that was below normal. So again, at this point, I think it's an open question. The physical removal of cells using the LRS does seem to be an important cause of this lymphopenia, but we don't know yet if it's the full story or not. There are a couple different ways to try to sort this out. I'm not going to spend really much time on this except to say that there's a group in Sweden that uses a large database now called Scandat3, where they get information on essentially everybody in the country. So the Sweden, like many of the Northern European countries, is really, really organized. Everybody gets health insurance through the government, and everyone's tracked carefully in a database. And so they were able to connect their database where they have information on thousands and thousands of platelet and plasma donors, every donation that they made, and they were able to connect that to health databases and try to see if they could see a signal for adverse events on an excess, for example, of immunocompromised, immune-related infections or bacterial infections, that kind of thing. they think they saw a bit of a signal in frequent platelet donors. They also use the treatment instrument mostly in Sweden, but it's very hard to sort out. There are very few really high frequency platelet donors in Sweden. So I'd say this is not kind of a definitive signal of harm, but rather something that merits some additional follow-up. And is anyway, I would say you can say that it's hypothesis generating, but not really definitive data. Anyway, so further work's being done on that. In the meantime, I started organizing a study called the Best PAL study. So this is with the BEST organization. Both Claudia Cohn and also Dave McKenna from Minnesota are part of this group. And what we wanted to do was to answer several of the outstanding questions from these initial studies. So first, we wanted to see if we could sort out the effects of age and platelet donation on lymphopenia among these donors. Second, we wanted to do a direct head-to-head comparison of donation on the Amicus instrument and on the treatment instrument to really define what the issues were there, and if there really was an effect of donating on an instrument that does not have this leukocyte trapping system built in. We also wanted to look longitudinally at these donors. So to this point, all we had was a snapshot at a single point in time. And finally, we wanted to look at immunoglobulin levels, which we hadn't looked at yet. So for this study, the plan was to get 131 frequent treatment donors or 131 frequent Amicus donors and compare them to a third group, which were frequent whole blood donors, 262. And these would be matched for age and sex. At baseline, the plan is to collect CD4 and CD8 counts, as well as get CBCs with diff and also measure total immunoglobulin. And then the plan is just to move forward in time and at different points over the course of 1 year, see what the counts were doing in the frequent platelet donors on either of the platforms. And the study had gotten off to a great start, was interrupted by COVID. But anyway, so here I'm showing the primary outcomes. So for baseline, we want to understand the central tendency and dispersion of CD4 and CD8 counts in the frequent treatment donors versus the frequent Amicus donors, compared with sex and age matched whole blood donors, and also look at the prevalence of PAL in the frequent treatment and Amicus donors. And then over 12 months, look at the CD4 and CD8 trajectories in the frequent TREMA versus the frequent Amicus donors. And these are the secondary outcomes. So we wanted to look at IgG in these platelet donors versus the age-matched controls, to look at the 12-month prevalence of PAL and its discordancy with the baseline prevalence in frequent TREMA versus Amicus donors. And then also finally look at the risk of PAL as a function of donor age, of lifetime platelet pheresis donations, of whether people were giving singles, doubles, or triples when they did their platelet donations, of the total platelet yield over the last 5 years, and look also, again, at the effect of which instrument was used. These were the sites that were and are participating in the study. I should say this was just launching right in the spring when things got bad in Boston in terms of the COVID pandemic. So it's currently on hold for the US. Canadian Blood Services, site number 1, had accrued all of their donors. So we asked each site basically to get 20 or 30 frequent donors. And so that was done both at Canadian Blood Services and in Australia. And so those two places are going forward. And we should have the 1 year data by the end of this calendar year. For the other sites, we're just going to wait until things settle down with the pandemic, but I hope to complete this study. And then really, I think the essential question is, do lymphopenic platelet donors have normal immunity, or is PAL just a lab finding? And so to try to answer this question, which is a really hard one, we're planning to do a vaccination study of frequent apheresis platelet donors. So here are our same 3 groups, 1 to 2 donations per year, 3 to 19, or 20 to 24 donations per year. And the plan is to vaccinate these individuals with something they haven't seen before. And so we intend to give them a vaccine for meningitis, which is now recommended for college students. And it's actually a control that's being used in at least one of the RCTs being done to look at COVID vaccines. So we will vaccinate these people and we will look for the immune response. The particular vaccine that's given is a quadrivalent vaccine. And so you can look at the immune response specifically to 4 different antigens, A, C, W, 135, and Y, as shown here. This was a study done by my collaborator, Lindsay Baden, and shows you that just some data in stem cell transplant recipients where it is possible to look individually at these 4 different antigens. In this study, you can see the dotted line going across the bottom. So a titer of 8 is thought to be reflective of immune protection. And so anyway, I think this will go a long way toward answering the question of whether this is something we really need to be worried about clinically, or whether it's just an interesting but totally meaningless lab finding. I will say that the treatment instrument and frequent platelet donation is something that goes on all over the world. It's been going on for many years. I have to believe that if this was causing widespread immunodeficiency and disease, it's something people must have noticed a long time ago. But it is possible, I think, that there may be a subtle immune defect in some of these individuals. I don't think we can really say. It is thought that something like only 2% of lymphocytes actually circulate. Most are in lymphoid tissues, lymph nodes, spleen, so on. So the exact peripheral count may not really reflect what these individuals' immunity is really like. But we'll just have to see. So in conclusion, frequent platelet pheresis on the treatment instrument is associated with severe lymphopenia. Donors with CD4 positive T lymphocyte counts less than 200 were all above 55 years old, and they all had 170 or more donations lifetime. Severe lymphopenia may persist after discontinuing donation. The lymphopenia mechanism is unclear. Many lymphocytes are sequestered by the TREMA LRS chamber. 20% of frequent amicus apheresis platelet donors, though, using a system that doesn't have an LRS, did have low CD4 T cell counts. There's no evidence at this point, at least, that the cytopenias are harmful, but it is something we're concerned about. So I have a lot of people to acknowledge, but mainly I'm John Gansner, my initial collaborator from the Brigham, Andy Romani, who's now working in Halifax, Nova Scotia, and Lindsay Baden, who's an infectious disease guy at the Brigham. So let me stop there. I'll stop sharing my screen, and I'm happy to answer any questions.
Thank you so much for that talk, Rick. It was fantastic. Can I open up the floor for questions?
Hi, this is Chris Hoquist. I'd like to ask a question.
Sure.
Do you think that the T cell loss is random with TREMA, or do you think there might be situations, for example, like infection, that either enhance or reduce the likelihood that T cells, for example, might physically interact with a platelet and then be trapped?
Yeah, I think it's a really good question. I think my working hypothesis is that there's not a selection. That is that we're kind of looking at a bulk T cell loss, but I don't think we know. That was not something that we've really explored, but I think it's a reasonable hypothesis. Yeah, so that's all I can tell you.
Rick, have you gone to the group of 20 and just done a check as to whether more of them have succumbed to infectious diseases?
Well, so that's the thing. We did a brief health screen. The problem is that if you can donate platelets, then by definition you're healthy. So I think that there may be a selection bias. So if there is, let's say, an adverse health effect of this, then I think that the way to find out is not by looking at necessarily at the people who are able to donate. It was one of the reasons why we wrote letters to the people who had stopped donating, and we asked them. The numbers were too small; like we couldn't. It wasn't possible to see any kind of signal. But yeah. So.
Yeah, this is John Crossan. That was a very interesting discussion. And I'm sure I just wanted to point out, I'm sure you're aware that patients who are HIV positive and actually clinically have AIDS will actually often have elevated gamma globulins in their serum. But of course, they don't make specific antibodies. And so I think your answer will come from the vaccination study. And my guess is that they will respond very well to vaccinations.
That is my guess as well. I think it's the best experiment we've been able to come up with to try to figure out if this is meaningful or not. Yeah, my guess is they'll have a normal response, but I look forward to getting the data eventually.
Hi, this is Steve Jameson. I think we've been in email contact before.
Oh, yeah. Hey, Steve.
Good to see you. Just one quick question. If there is a form of lymphopenia-induced proliferation being induced, have you had the opportunity to take samples close to the time of donation and then progressing from that to see if there's actually an increased frequency of memory cells over time in the blood, memory phenotype T cells in the blood, following the lymphopenia? So it would be kind of on the rebound that you'd get an even higher frequency of memory cells.
It's something that it's something we want to look at, but we haven't done it yet. But yeah.
Any other questions?
I have a question. This is Bartek Grzywacz. Have you looked at other lymphocyte subsets? Natural killer cells are not timeless dependent, so the question would be, are NK cells affected? Are B cells affected in a similar way?
So let me go back. I don't have sort of the detailed data in front of me, but I believe though that there were similar findings with the NKs. Let me just look really quick. So yeah, sorry, I don't have the data in front of me. We mostly focused on the T cells. Part of it was just sort of amount of sample that we had and amount of money that we had to do it.
And, you know, in part, it's a selfish question because myself and others that I work with use trimacons for research to isolate NK cells for in vitro studies. So it would be important to know because this obviously could be a factor in the results.
No, and it's a huge factor here. We provide, you know, I don't know which, I imagine, Claudia, do you guys use the same instrument?
We don't have a donor center here, Rick. We depend on JED and the Red Cross.
I see. So yeah, no, we provide these cones routinely to investigators for exactly that purpose. Yeah, so let's see. I don't know. I'm gonna have to look through our supplemental data. But if you want, feel free to send me an email after, and I can let you know what else we found.
Yeah, I'll be glad to. And regarding vaccination, I just wanted to mention that we know, I mean, it's very basic immunology that I'm at my level, and that there are T-cell dependent antigens and T-cell independent antigens. So I'm not sure the meningococcal vaccine, which one is it, but it may be polysaccharide based. It may be actually T-cell independent antigen.
Yeah, I was told that this one is thought to be T-cell dependent. But I don't know, maybe Steve can comment on that, but that was what I was told by our ID folks.
Anything else? If not, I want to thank everyone for attending this talk, and thank Rick most of all for presenting this today.
Oh, thanks. Well, my pleasure.
And everyone, please hang fire while the votes come in, and we'll know what the result is by Friday, I guess.
All right. Thanks very much.